Oh, no? Okay. Integumentary system, is that right, guys? Is your skin, and the function of skin is protection. The first line of your immune system is that better now? You can see it a little bit better. I hope it's in the video a little bit better as well. So let's go on. We should make these videos as short as possible. Protection, conified, stratified, squamous epithelial tissue on the very top layer. Is that right? Your epidermis. There are 3 layers in the skin that you must know. One of them is epidermis, dermis, or they call it eodermis, and hypodermis, or subcutaneous, or subcu, some people call it for short. Please call it subcutaneous. Don't use any short. The top layer, what tissue it is made up of? It is made up of cornified, stratified, squamous epithelial tissue. It's more than 1 layer.
Can we say horny as well or no?
Horny, that's fine. Yes. Okay. And
so we can just switch between hypodermis and the other one, right? And it's still be
hypodermis and subcutaneous, same name, same thing. We are crazy in biology. For one thing, we call it 2 different things. Same thing, but we call it different things. It's like you call me Amir and George. I say, sure. That's how crazy biology people are. No, it all has a history behind it. Why they call it 2 different things. Originally, somebody proposed it, and somebody else came and called it something else, and everybody followed it. Everything has a reason in biology. But we have a lot of terms like Volkmann canal and perforating canal, same thing. Central canal and Herbertian canal, you remember that? Same thing. But anyhow, right here is stratified, cornified, stratified squamous epithelial tissue. Underneath is your dermis, made up of dense irregular connective tissue made up of collagen. And underneath of that, it is your hypodermis, made up of adipocytes. Okay, very 3 layers of the skin you should know. Dermis is a true skin, and it came from mesoderm. You don't know what mesoderm is yet. You will, next exam, we will talk about mesoderm. Dermis contains connective tissue, blood vessels, pigments, and not the fat cells. Keratinization, quickly, to refresh your memory from your other classes, the very bottom layer of cornified, stratified, or keratinized, stratified squamous epithelial tissue are made up of cells that they undergo mitosis, and they divide, and they push the cells upward. Okay, so when they're dividing, the very bottom layer here, when they're dividing and they push the cells upward, when they reach a certain level up here, they die. And when they die, the nucleus dies, the cell dies, and what is left of it, it is keratin, the protein molecule keratin. Okay, so that protein molecule, you have dead skin on top of your body. Right here is all dead skin. You can put it on a microscope, take a look at it. It's all keratin. Okay, so that's what the name of the process is called, keratinization. And that's one of the questions at the beginning of the lecture. You remember that? I hope somebody bring up those questions, so I make sure that I answer all of the questions that pertains to today's material. Okay, and subcutaneous layer is a very bottom layer of the skin. Here is they're talking about exoskeleton in arthropods, which we'll talk about it later on. Don't worry about it for this exam, but these are the layers of the skin. As you mentioned it, you said it, the hair follicle and erector pili is in the dermis layer of the skin. This is epidermis layer, dermis layer, and subcutaneous on the bottom. The hair follicle is made up of smooth muscle. It means you do not have control over it. This is one of the places that is not involved with the tube in our body. You remember last time I said smooth muscle, wherever you have a tube in your body, most places you have tube in your body, that's where smooth muscles are. But this is one place that there is smooth muscle and there's no tube. They attach to the hair follicle, and whenever you're scared or you're cold, they contract and give you goosebumps. Other emotional type of thing gives you goosebumps. You watch a sad movie, whatever, that gives you goosebumps. In other animals, when they are into a fight stage, they erector pili contract, so the hair extend, becomes like this, so the animal is shown, I'm big, I can fight you, or something like that. It's just a defense mechanism toward the whole entire animal. Anyhow, the pigments in the skin, there are a variety of them. One of them is melanin. Melanin is the most common pigment, which is black or brown pigment in animal kingdom. Okay, we do have a lot of melanin in our skin. Carotenoid is a yellow, orange. This is yellow, xanthophore, iridophore. Iridophore type of pigment contains crystals of guanine or other purine. What is guanine purine from Bio-1?
It was nucleotides.
They are nitrogenous bases. Very good. They are nitrogenous bases. You found them in DNA, RNA, and then they are the ones that allow animals to camouflage themselves, to change their color, like octopus. Okay, they can change their color because of these pigments in the skin, and the mystery is how do they recognize their environment and they can change their color to the environment they are surrounded by. That is something we still do not have a good grasp of it as far as biology goes. As far as skeletal system, so I'm all done with skin. If there is I explain keratinization. If anybody have those questions, and before you, I explain the structure of skin. The name of the system is integumentary system. First line of defense, prevent bacteria, viruses, other things into our body. And then, of course, I talked about the keratinization as well. If there's anything else I'm supposed to explain, please let me know. The next one is skeletal system, provides supports. Exoskeleton, something we will talk about it maybe in May, or late April, May, when we get to arthropods. Endoskeleton, something we have. Our skeleton is inside of our body. Our bone is not outside of our body. And arthropods, you will see the bone is outside of the body. That's what they call an exoskeleton. Okay. Calcium and phosphorus, we have them. The endoskeleton, the exoskeleton, you will learn that it's made up of protein and chitin, something different than ours. Ours is mainly made up of calcium, phosphorus, and other protein molecules. Chondrocytes are, chondro, it means cartilage. They are bone cell. They are cartilage cells which eventually become bone cells. If time comes up, I will talk about it. Hyaline cartilage, you do have a slide of it. Just recently I put it in. It's not on DocuShare or on my website, but it is. Different types of bone development, endochondral bone, rest of the body, and intramembranous bone, the skull. When a baby is born, between the plates of the bone in the skull, they call it sutures. Those are membranous, intramembranous type of cartilage, and over a period of time, they become tough, rough bone. But the rest of our bone, the rest of our body, the bone becomes a hard bone from, they call it endochondral bone. At the end of your long bones, right here, mine is ossified now, it's all bone now, but some of you still growing. At the end of your bones, right here and here, there is a plate of cartilage, and that becomes long, your hand becomes longer and longer. If you have a baby at home, that's what they are at the end. And that's what endochondral bone they are. So at the end of your bones, they are cartilage that they are growing and growing and growing, and gives the person length. Anyhow, so this is pretty much the histology of the bone. As I said, blood vessels comes into the bone, right? This is one of your long bone. We have 3-4 different type of bone: irregular bone, flat bone, irregular bone, okay, long bone, okay. But don't worry about those. So the blood vessel comes in, into the bone, and then of course this is the bone marrow, the soft portion of the bone. This is the compact bone, and the compact bone is made up of this, look like this that I showed it to you guys. This is the compact bone, right? And we talked about it. Made up of many osteon. The osteon are connected together by perforating canal. Central canals are connected together by perforating canal. And when food and nutrient from blood vessels, either perforating canal or central canal, they go through canaliculi, these little hair-looking, do we have a bigger of it? Yeah, these are canaliculi, the line right here. All of these lines, like a spider, legs of the spider right here, they connect blood vessels to osteocytes, from osteocytes to the osteocytes, food and nutrient, because this is right here, guys. Remember, this is the matrix of the bone. That's the hard calcium phosphorus. No food and no nutrient, no blood can go through here. So what happens, food and nutrient through canaliculi go to this osteocyte, and waste material from this osteocyte go into eventually the vein, the venules, in the central canal, perforating canal, and they leave bone, because you do make waste material in the bone as well. Your bone is remodeled every 6, 7, 8 years. So you're constantly destroying your bone and making up new bones, constantly. And I will talk about that hopefully in a minute. Okay, here are the bones that you should know. They broke down your bone to two different categories, axial bone and appendicular bone. Axial bone, I put X mark on them, which is the skull, the bones of the skull, and the bones of the ribs and spinal cord. These are called axial bone. Appendicular bone is any bone outside of these bones, like your bones of your legs, arms, all of these are they're called appendicular bone. And I'm hoping you would know the name of those bones for your lab practical exam. And also could be a lecture question, which one of the following bones, or quiz question next time, which one of the following bone is an appendicular bone? Which one of the following bone is an axial bone? You should know that. Stick to this. I know you took anatomy, you know a lot, you know more than I do, but stick to this, okay? The other bones that your spinal column, that I would like your vertebral column bones that you should know, the very first bone right here on your neck, the area of your neck is called atlas, and the second one is called axis. Okay, atlas is the bone that allows your bone to move front and back, okay? And the second one, the axis, is the bone that allows your bone moves to the right or left. And then the bones in the neck, I'm hoping you will know those, it's called cervical bones. The bone in your chest area is called thoracic bone, and the bone in the back area is called lumbar bone. And then of course you have the sacrum and coccyx, your tailbone. Some people are born with a longer tailbone. Right after you're born, the pediatrician, I don't know who does perform the surgery now, cut the bone, the tailbone, to normal size, so the baby does not have to sit. That's one of the things we evolved. We got rid of our tail bone. Most animals have tail. We got rid of it. And the good thing, I'm talking about evolution. When we got rid of it, then we can sit down. That's an advantage. Other animals cannot sit down. When we sit down, then we can use our arm. That's another, we are upright. Other animals have to walk with all four of them. We can walk with two of them. And the other one, we can use these hands. Imagine horses could sit down, but still they cannot use their hands like the way we can use them. Okay, so that's all of that goes back to evolution. Make sure you know whatever I say about evolution throughout the semester. Make sure you know them a little bit. It could be a quiz question, an exam question. Jose, you had a question a century ago.
Yeah. Could you go back to the last slide?
Last slide. Oh, okay.
What was the difference between axial and
Okay, axial bones, usually the bones that are in x-axis,
Okay.
they call them axial bones. And what bones there, this is not in x-axis, it's in y-axis, right? So whatever bones are in x-axis, they call them axial bones that are the skull, the bones of the skull. There are a few bones in your skull. And the bones of your ribs, okay, and your vertebral column. They're all referred to as axial bone. So your cerebral bone right here, your cervical bone right in your neck area, it is part of the axial bone. Your phalanges, right? Your phalanges are part of your appendicular bone. Study them a little bit. Okay, let's move with bone. Cancellous bone is spongy bone. You saw that. So all of this area, right? This area. This is compact bone. This is spongy bone. The spongy bone is another name for it is cancellous. Biologists are crazy. For one thing, they like to call it two different, sometimes three. Sometimes there are three different names for one thing. I don't know. I didn't make it up. I didn't make it up. None of these things you see, I made it up. It's all out of my hand. Farhad, long time ago.
How did you, when you were an undergrad student, how did you study? How did you?
Repetition. I read my notes every night from beginning to the end.
Okay.
Yes?
Is the spongy bone, does that bone have the bone marrow, or is the spongy
That-
bone and bone marrow two different?
No, okay, good. Okay, you see all of these blood, your blood is made in your bone marrow, right? So your bone does not stop, your compact bone does not stop all of a sudden and become bone marrow. So slowly, you still have some osteocytes here. Slowly, then it becomes your bone marrow. And the very, very, very inside of your inner portion, you don't have spongy bone anymore. It's just bone marrow. So it's a transition. Very good. It's a transition between compact bone and bone marrow. It's a spongy bone. So in spongy bone, you do have blood structures, and they are making your stem cells are in the spongy bone as well. Okay. Compact bone, osteoclasts are the bone reabsorbing. They are the cells in the bone that break down bone. Okay, reabsorbing they call it. And then osteoblasts, they are the bone, easy to memorize, building. Osteoblasts, they are the bones, they are the cells that they make up your bone. So as I said constantly, your bones are being remodeled. They break down by help of these cells, and they are building up, your bone is being built up by these cells. And these cells are right up here, right up here. And some of these osteocytes are osteoblasts, some of them are osteoclasts. Calcitonin, it lowers the level of the calcium in your blood. So if you have too much calcium in your blood, the hormone calcitonin that is secreted from your thyroid gland, your thyroid gland looks like a butterfly, right here secretes calcitonin, and calcitonin goes to your blood, throughout your blood vessels, and they take the excess calcium and bring them to the bone. And when they bring them to the bone, what happens? Which one of these two cells you think needs needs calcium? Sorry. Which one? Osteoblast. So calcitonin works with osteoblast to build bones. Am I making some sense a little bit? Then the other hormone that comes from your parathyroid gland at the back of your thyroid gland, if I turn around, there are 4 parathyroid glands. They are separate, but in association, they're close to each other, to thyroid gland. They are parathyroid gland that makes the parathyroid hormone and increases the calcium level. So which one of these two you think parathyroid works with? It works with osteoclast. It tells osteoclast to start working to break bone down because calcium level in your blood is what? Low. You need to bring your calcium level, the calcium level in your blood needs to come up. Our body can sense that. There are receptors in your cells and so on and so forth. That's cell biology material. We're not getting there. You studied the cell receptors in Bio 1, I hope. I know you did, Dr. McRae talks about it all the time. Okay, so let's go. Muscular movement, I'm done with bone. Is there any question on the bone I did not address? At the beginning, remember, I talked about how bone is being remodeled. I talked about bone structure, so on and so forth. Is there anything else? Jose, yes.
Are osteocytes matured osteoblasts?
Huh? What?
Are osteocytes matured osteoblasts?
No, osteocyte is a generic term. And then we have osteoblast and osteoclast, which they are a different type of osteocytes, which they build up bone and break down bone. Am I answering your question?
Yeah.
Okay, that's it. Yes.
So would bone marrow still be osteocytes?
There are some, yeah, sure. In the bone, not in the bone marrow, in the spongy bone. No, I'm sorry. In the bone marrow, you don't have actual bone marrow. You do not have any more osteocytes. You have stem cells that can possibly become osteocytes. But the spongy bone portion, you do have osteocytes. And then you have a little bit of bone marrow. As we said, it's a transition to bone marrow. Okay, I don't know, next time when you go to Kentucky Fried Chicken or something like that, you get a bone and crush it with your teeth. You can crush it. Some bones, some don't do them. You break your teeth, but you crush them, and you can see actually the spongy bone and the bone marrow. A lot of people eat that bone marrow. I do not recommend it, but when I was a kid, I used to eat them all the time. But now, because of all of the antibiotic, pesticides, herbicide, all of that, I will not recommend it. You're eating a lot of, I think, poison, if you do eat it. Don't eat it. Liver, don't eat it, please. I think it's all no good. But anyhow, I hope I'm not scaring you. Sliding filaments model, they talked about it a little bit, and I will go over it. Just hang on. Atrophy, when the muscle cells will die, it's called atrophy. You don't use them. You don't lose them, you lose them, something like that. There's an expression, use them or lose them, don't use them, you lose them. Eric, you know what I'm talking about. No? Hypertrophy, it means the size of the cell increases. When you exercise, when you do bench press, and so on and so forth, your bone undergo hypertrophy, it means the size increase. Hyperplasia is when the numbers increase, and that doesn't happen to you when you exercise. Unless there's a cancer, God forbid, if there's a cancer, then your muscle cells undergo hyperplasia. So when you exercise, your muscle cells undergo hypertrophy, the size increase. There is a concept in biology, guys, we call it utility. Utility, it means constant number of cells. You were born, all of you in here, you were born with a set number of cells, neuron, in your central nervous system, and a set number of muscle cells, both smooth muscle, cardiac muscle, skeletal muscle, so on and so forth. And you will die with those numbers. They will not change in your lifetime. Am I making some sense? But the bones of your, the cells of your bones, they will change. You were born with, let's say, I'm making up a number. Let's say you were born with 1 million osteocyte. Now you have 10 million osteocyte. When you get old, you have 5 million osteocytes. Am I making some sense? That's not true about your neurons. I'm talking about a normal person, a person that has a healthy life from time of birth until it dies. Am I making some sense? That's not true with your neurons. That's not true with your muscle cells. And that concept in biology is called utility, constant number of cells from birth to death. Oh, yes, no, maybe perhaps. Okay, so, so for muscle cells, in normal situation, you undergo hypertrophy. If you exercise, you lift it. Attack pull release, another name for sliding filament, about 50 to 100 of them. Keratin, you remember the question today, Rod is going to exercise, Alex or whoever it was, well, he possibly needs to take keratin. I do not recommend it, but now it's legal. We don't know. Athletes can take keratin as a shake, but later on, I don't know, 10 years, 20 years, 30 years, it's been in the market for the past, I don't know, 15, 20 years, and it's not illegal yet, but it could be like steroid. At the beginning, it was legal, but now they saw the devastation on the steroids, they said it's illegal now. So keratin, we don't know what effect it has on the body, but people are taking it. I guess it will help. It's a source of, it stores ATP, allows the ATP to be stored in the cell. Red muscles are highly vascularized and have many mitochondria. So one thing you do when you exercise, you're increasing. We have to make the recommendation to Rod to exercise to increase the number of mitochondria. You cannot increase the number of the muscle cells, but you can, by exercise, you can increase the number of mitochondria if you exercise. Okay, so here it is, inside of your muscle bundle, just like nerve bundle. Inside of muscle bundle, you have muscle cells, and you have the H zone, I band, and so on and so forth. I think it is explained in some of the model. It's not explained in any of these guys. But anyhow, let's go over it quickly. So you have these darker line, which you see on the microscope. It's called myosin. And then these lighter bands is called actin, or thin filament. Myosin, or thick filament. And they are attached, the actin is attached to Z lines. Right here is Z line, Z line. From Z line to Z line is called a sarcomere. That's just the definition of sarcomere. From Z line to Z line is called sarcomere. And then, of course, these are nucleuses on the periphery of the cells, and so on and so forth. Okay, then by definition, I band is where there is only and only actin filament, or thin filament. A band, look from here to here, is called A band, which there is both actin and myosin, by definition. Okay, and H zone, right here, where is H zone in this? That's a Z line, H zone.
Right on the bottom.
Is right here, H zone. I think this is wrong if this is Z line. But H zone, by definition, H zone, right here, thank you, on the bottom. H zone, that's where there is only and only actin. So you have one area which is only actin, myosin. H zone is where there is only myosin. You have one area that is only myosin, and that's H zone. One area which is only actin, which is what? I band. And then one area which you have both, A band, you have both actin and myosin. You have both actin and myosin, is called A band. Okay, so having said that, which you should know a little bit, here is the myosin, the head that I was talking about, tail. All of these head and tail come together in the myosin area. Right here, myosin. And then, of course, this is the structure of the actin. Actin, you have actin molecules, like here, the tennis balls. The tennis balls are connected together by a rope called tropomyosin. And every so on and so forth, in the tropomyosin, you have the molecules called troponin complex.
Now, the reason why
Okay, maybe I can answer it here, Farhad. So when, right here, a neuron, I should have, and the same as this model, when a neuron releases neurotransmitters at axon knob, and this is muscle, right? Everybody can see that. This is muscle. When neuron releases the neurotransmitter, acetylcholine, and acetylcholine gets to the T tubules in here. Again, I'm not going to go into details of it too much, but it goes, attaches to the cell membrane, put it this way, of the muscle cells, then allows the sodium channels open up. Remember the sodium channels from last lecture?
Yeah.
From one neuron to the next neuron to the next neuron. So the sodium channels open up, there's a whole bunch of sodium, and all of the sodium goes inside of the muscle cells. What is the name of that process? Action potential in where? a muscle. Same thing, they call it same thing. You're lucky. They didn't call it something else. They didn't call it Amir potential. They didn't call that. They call it action potential, of course, in muscle cells. Okay, so when a lot of sodium gets into the muscle, then those sodium causes calcium to be released from sarcoplasmic reticulum. You guys see that? Sarcoplasmic reticulum stores calcium in the muscle, stores calcium in the muscle. We do have another one right here. If you look at it, here is the neuron, here is the neuromuscular junction, and axon knob, right, innervating skeletal muscle. Okay, but anyhow, calcium is released from, because there is so much sodium inside of muscle cells, the calcium is released from sarcoplasmic reticulum. They come and sit on troponin complex. When they sit on troponin complex, it changes the shape of the actin, and then the head of the myosin attaches to actin molecules, to the tennis balls. These are called ping pong balls. The troponin complex are called ping pong balls. And then, of course, the head tilts. I'm going to make some sense. Tilts a little bit, and then releases itself and attaches to another portion. Tilts, and that's why you can contract your muscles. Your muscles shorten. When you contract your muscles, what happens? Your muscle shortens. I hope I'm making some sense. And then, of course, it releases, attack, release, attack. And for every detachment, you need ATP. Detachment, not attachment. Detachment, you need ATP. We talked about that. Here again, same picture, same thing. Here is a summary of what's going on. I'm not going to go into the details of it. And at the end, one thing I should say, at the end, all of the calcium here must go back to sarcoplasmic reticulum. And you need ATP for that as well. Okay, so the calcium here, the excess calcium here cause muscle contraction, and got to go back to sarcoplasmic reticulum at the end of the day. Not the end, at the end of the muscle contraction. Sliding filament, I think with atrophy, we talked about it. Hyperplasia hypertrophy, attack for release. So since you need a lot of ATP in your muscle contraction, there is naturally creatine in your muscle cells that attaches to ADP, and then another phosphate comes to it, it becomes ATP, and then ATP is needed for detachment. That's how you have a great source of creatine in your muscle cells, and athletes are trying to increase that so they can increase the amount of ATP eventually that they need. I hope I'm making some sense. Red muscles, high vascular portion, many mitochondria, white muscles, so on and so forth. There is some other thing I forgot to mention on muscle contraction, but anyhow, if it comes up, we'll talk about it. Okay, the next one is your blood, intracellular. A few definitions, I wrote them down for you. Intra, I guess it was in your prefix suffixes quiz last time, but intra, it means inside a cell. Intra. Inter, you call it interstate, or do you call it intrastate? If you go to the gas station, you say intra 99, highway 99, you will not understand. But if you say interstate 99, that's wrong. Because 99 is a freeway or highway 4. 99, I've seen it in Canada, 99, they call it 99. But in highway number 4. Number 4, highway 4 is intrastate freeway. It's not, it is intra, here you go, I'm making a mistake. It's intra freeway. It means inside of the cell. Inter, which is inter, it means between the cells. Okay, intracellular, inside of cell, extracellular, there are two types of them, intercellular and plasma. Intercellular or interstitial fluid is right here. Well, that doesn't work. That works between tissues, I should mention that. And then you have plasma. If I take your blood and put it in a test tube, let us sit down or spin it. If I spin it, the solid portion of the blood goes down, then you have a thin layer between the plasma of the blood, the liquid portion of the blood, and then between the solid portion of the blood is called a buffy coat. Plasma is the liquid portion of the blood. These two together, the buffy coat, which is your white blood cells and red blood cells on the bottom, and the and other solid portion is called form elements. Here is a blood smear. You do have slide of that. In the blood smear, you have 3 different type of white blood cells: basophil, neutrophil, the 3 musketeers. Okay, this is a basophil. Look at the granules. Don't look at the nucleus. This is the nucleus. This is the granules, right here. The granules are dark. That's what they call them basophil, because when they stain them, they pick up the base. You know, acid base in chemistry. We were talking about that today. This one, of course, nucleus is purple. Don't go by shape of nucleus to identify the cells on the microscope. I put them up on the microscope here today for you. And then this is eosinophil. The granules are orange-red. Okay? And then neutrophil, look at the granules. They do have granules. These granules do not pick up acid or base. This picks up acid. They call it acidophil. Phil means what? Love, like. So they love acid. They pick up acid when they stain them. The neutrophil does not pick up anything, not acid, not base, but they have granules. And you do have a lot of these under your microscope, so I will not put it on demo today. I just put these 2 on demo today. They are, all 3 of these 3 musketeers, they are about 2.5 times of red blood cells. About 2.5 times. Okay? Then we have 2 other white blood cell, lymphocyte and monocyte. Okay? Lymphocyte is same size as red blood cells, almost. You can look at it on a microscope. You do have them in your slides. And the cell is filled with nucleus. Look at the nucleus, how big it is. It fills up the entire cell. And when you have humoral immune response, these are the guys who respond. Okay? I did make a chart. Yes, we will show. This is monocyte, another one that does not have